Welcome my dear students. Today I am discussing about Williamsonia sewardiana. So now in your screen there is a plant. It is none other than Williamsonia sewardiana. Okay. The genus Williamsonia was first discovered by Williamson in the year of 1870 from the Jurassic of Yorkshire, and he named the fossil as Zamia gigas and placed under the order Cycadales. Who was Williamson? He was the grand teacher of Birbal Sahani. So he discovered the genus in the year of 1870. But due to the presence of flower-like fructifications, it was placed under the order Banatitles. It means that Williamson placed under Cycadales, but it was then later on transferred to the order Cycadioides due to the presence of flower-like fructifications. So, in the both above said order, the leaves were similar in form, similar in structure, and similar in venation, but there is a difference. What is that? They differ in stomata, that is haplochelic stomata in Cycadales, but syndictochelic stomata in Cycadioides, that is in Banatites, yes, or we may say in Williamsonia the stomata is of syndictochelic. And now I am giving you a special point for MCQ. It is that the two order coexisted during Mesozoic era from Jurassic up to the Cretaceous period. So this period was known as the age of cycads. So a question may arise, which period was termed as the age of cycads? You will say that the Jurassic to Cretaceous period of Mesozoic era is termed as the age of cycads. In the next page, it is written that 5 Indian species of Williamsonia are. It means that in India 5 species were discovered from the Rajmahal hill of Bihar, and these were, or we may say these are, Williamsonia indica, Williamsonia microps, Williamsonia shahani, Williamsonia santalensis, and Williamsonia sewardiana. That is, India represents 5 species of the genus Williamsonia. Now we have to discuss about Williamsonia sewardiana. Birbal Sahani in the year of 1932 reconstructed the plant, and the study material were collected from Rajmahal Hill. Why it is termed as, already is designated as the reconstructed plant? Because in this case the leaf genus, the stem genus, the male fructification, the female fructifications were fossilized separately, not as a whole like a plant. The different parts of the plants, that is the stem, leaf, male fructification, female fructification, they were fossilized separately and in separate places. So these were collected, these were studied morphologically, anatomically, cytologically, and after these critical studies it was opined that they were from the same plant. Hence Birbal Sahani in the year of 1932 he reconstructed the plant. These were the fossils belonging to the Jurassic Upper Indian Gondwana. If the question question is like that: name one Gondwana plant or Gondwana flora. You may write down Williamsonia, sewardiana. The plant was named to honor Professor Williamson. He was the grand teacher of Shani, and Professor S.E. Seward, he was the direct teacher of Shani. Okay. Now, what are the form genuses of this plant? The leaf genus was termed as Tilophyllum Cutchans. The stem genus was termed as Bucklandia indica. The male fructifications was termed as Weltrichia, and the female fructification none other than Williamsonia. Now we are moving to the third page. Here it is discussed about the morphology of the plant. I am discussing point wise. The first point: the plant consists of branched, slender, columnar trunk of about 2 meters long. The trunk, that is the stem trunk, the main trunk is 2 meters long. There are prominent spirally arranged rhomboidal leaf bases and unipinnate crown of compound leaves. That is, the whole trunk is spotted by the leaf bases. The stems are simply monopodial or dichotomously branched, and the branching is lateral and project through the thick armor of the leaf bases. And regarding the branches, these are of two types: sterile of vegetative shoot, that is, they didn't bear any cone or flower, and the fertile, which bears cone or flower. Similarly, leaves are of two types: the simple scale-like and pointed leaves on the main shoot, and pinnately compound foliage leaves on the lateral shoots. And the main stem, it is to be noted that it bears both types of the simple scale like leaves as well as the pinnately compound foliage leaves. Now we are moving to the fourth page. It is about the stem anatomy. The transverse section of the trunk revealed a circular outline. It will be circular. The cortex is white, parenchymatous without secretory sacs. It means that the epidermis is absent, that is, it was not fossilized. The pith is narrow and it is made up of parenchyma cells with secretory sacs and extended between the vascular bundle as rays. That is, pith extends through the vascular bundles. The primary vascular bundles are enlarged, conjoint, collateral and arranged in a ring-like fashion. The cambium is clear. A prominent ring of secondary wood is present around the primary xylem. That is, here primary wood as well as secondary wood is present. The secondary wood is traversed by both uni, bi or triseriate secondary medullary rays. The wood is monoxylic, that is compact, and scalariform tracheids with border pits are present. Multiserrate border pits are present on radial walls. Leaf traces numerous but without any girdle. Now we are moving to the fifth page. It is about the leaf, that is the form genus Tilophyllum. The first point is sickle shaped pinna are attached to the adaxial surface of the rachis by full, wide and round bases. That is, the both side of the rachis is having sickle shaped pinnately arranged leaves or leaflets. The venation is parallel. There are many leaf traces in a leaf base, because after arising from the primary xylem, it passes through the secondary xylem to the cortex, and where, that is in the cortex, it again branches in many times. Regarding the stomata, stomata is syndictochellic and arranged at right angles to the veins, and epidermal cells are wavy in outer walls. In the 6th page, I am discussing about the reproductive structure, mainly female reproductive structure. The genus Williamsonia is unisexual and dioecious. That is, male plant and female plants are separate. Only female plants of Williamsonia sewardiana have been discovered. Regarding the female fructification, we may say, number 1, here 2 figures are there in the corner. The upper one is the ovule and the second big one is the female fructification. Okay, the first point is, from the axil of the leaf cone bearing lateral shoot arises, and the shoots also bear both scale and foliage leaves. Number 2, there is a central conical receptacle in the female cone bearing stalked ovules, that is, ovules are stalked, and fleshy spirally arranged interseminal hair like scales. At the basis of the cone there are many perianth-like bracts. These bracts also have scales at the bases and hairs above. The tip of the receptacle is naked, that is, it is not covered by the bract. Each ovule is orthotropous and short-stalked. The integument is single around the nucellus. The tip of the nucellus extends to the micropylar beak and the pollen chamber. And the interseminal scales present among the ovules. The micropylar canal is narrow and lined by thin walled cells. 2, 3 or more archegonia are found at the apex of the endosperm. The embryo is dicotyledonous. So if we study the ovule, then we may see that the ovule is having a short stalk, having one integument, inside that one nucellus, which is at the apex. It has made the integument and micropyle and the nucellar beak, pollen chamber also, and inside that the female archegonia are present. In the 7th page, it is about the male fructification. The male fructifications were never found in connection with the plant. The form genus is termed as Weltrichia. The first point, the microsporophylls arranged in a hole and fused to each other at the base to make a cup-like structure. The second point is, the microsporophylls alternate with the bracts. Number 3, each microsporophyll band upwards and bears many minute appendages in the inner surface, each with 2 rows of synangia, and the pollen grains are monocolpate. Okay, so this is about the Williamsonia sewardiana. Hope this note will help you for your examination purpose. Thank you.